Hello, everyone, and welcome to today's webinar, Enumeration and Molecular Profiling of Circulating Tumor Cells for Diagnosis and Therapeutic Monitoring of Metastatic Cancer. I'm Shelley Mulock of Labroots, and I'll be your moderator for today's event. Today's educational web seminar is presented by Labroots and brought to you by Thermo Fisher Scientific. To learn more, visit thermofisher.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well, if you have trouble seeing or hearing the presentation. I'd like to now welcome our speakers, Dr. Pravin Potdar. Dr. Potdar received his Ph.D. from Tata Memorial Centre Mumbai, India, and has been researching cancer for over 30 years. For a complete biography of today's speaker, click their name in the presentation window at the top right of your screen. Dr. Potdar, you may now begin your presentation.
Yes. Good morning, everybody. I am Dr. Pravin Potdar. Presently, I am the chairman of the Institute Ethics Committee of Dr. Apj Abdul Kalam Research Centre Mumbai, as well as I am the former head and chief of Department of Molecular Medicine and Biology at Jaslok Hospital Research Centre Mumbai. I have been working in the field of cancer research for the last 30 years, and my present interest is studying the liquid biopsies containing circulating tumor cells and circulating tumor DNA for diagnosis and therapy monitoring of metastatic cancer. Today's my talk will be on enumeration and molecular profiling of circulating tumor cells for diagnosis and therapeutic monitoring of metastatic cancer. Before starting my talk, I would like to thank Thermo Fisher for giving me this opportunity to present my research work in this webinar. I'm also thankful to the special thanks to Lillian Manley, Jack, Julie Kevleker, then Megan and Israel for their continuous support in this matter. Coming to my first slide of today's presentation, as you all know that cancer incidence as per the Globicon 220, there are about 20 million people suffering from cancer and almost 10 million people are dying with the cancer every year in the world. The major reason behind it is due to the 80% diagnosed with this cancer at metastatic stage. And so far, it is difficult for the clinicians to monitor cancer therapy at this stage, resulting into death of this patient. If you see the general mechanism of cancer development, you know cancer is a multi-step process. It has got 4 stages. It has been well proved that if up to the patient comes up to the stage 3 level, they can able to get a proper treatment and clinician doesn't have to much think about giving the treatment. However, when the stage 4, the patient enter into the stage 4 stage, it is really very difficult for the clinicians to think because so far very few hospitals or clinics have the proper monitoring systems. It is already been seen that almost 70 to 80% patients are diagnosed at metastatic stage due to the lack of treatment or vigorous dose of chemotherapy or radiotherapy. Now, if you see the overall conventional methods of cancer diagnosis and therapy present situation, most of the laboratories, most of the hospitals which are in India, only metropolitan cities, they have the advanced technology. However, most of the people or all over the world also, I will say, mostly the cancer is diagnosed by the tissue biopsy, histopathological report, which is the final confirmation of the cancer. Once that is come, then clinicians plan the according to the stage, surgery, radiotherapy, and chemotherapy. However, monitoring of the therapy is randomly done by physician. Only they they do by appearing or history of the cancer patient, which may not be very much correct. And repeating biopsy is difficult in most of these cases because the patient is already in the very much great pain and he is not able to ready for the biopsies. And overall, the monitoring system fails and there is a great drug toxicity and many times it is fatal. So what is the alternative for this? Now, new concept of cancer therapy has been now introduced. That is called a liquid biopsy. Liquid biopsy is non-invasive method for testing the cancer, metastatic cancer at different stages of metastasis. Now, in liquid biopsy, today we are going to focus on the circulating tumor cells, which are one of the part of liquid biopsy and have great importance in cancer therapeutic. There are major two parameters which we have to see in circulating tumor cells. One is enumeration and second is a molecular profiling of the circulating tumor cells. Once we have the enumeration, we can define the stage of the metastatic, how easily we can control this metastasis. And molecular profiling of the CTC will give the exact genotype of the CTC, and we can target that CTC. And once we follow up this patient, we can have better cure than conventional methods. And this is already been following up for few hospitals. They are already doing this work and they are successful in doing this. Now, just to introduce targeted therapy of cancer for cancer, because this is whole exercise is mainly depend on giving the targeted therapies to the patient. So targeted therapies are the treatment that target specific characteristic of cancer cells, such as protein that allows the cancer cells to grow in a rapid or abnormal way. Targeted therapies are generally less likely harmful to the normal cells, healthy cells, than chemotherapy. Some targeted therapies are antibodies, whereas others are small molecules, which inhibits mainly the signaling pathways of cancer cells, which are responsible for their growth. In the right-hand side, you can see the targeted therapies. There are various inhibitors for the signaling pathways has already been introduced. Many targeted therapies compounds have been now discovered, and therefore you can choose the targeted therapy according to the genotype of cancer cells. Now, coming to the liquid biopsy and circulating tumor cells, which is our today's topic. Now, liquid biopsy protocol, if you see the liquid biopsy protocol for cancer therapy, it looks to be very simple, but there are various limitations in this, which we are going to talk today in my talk. Whole plasma can be collected as a liquid biopsy from the metastatic cancer, highly metastatic cancer patients. We can isolate the CTC or ctDNA. Today we are talking only on the circulating tumor cells, that is CTCs. After getting the CTCs, we do the enumeration of these CTCs. According to the enumeration, we can know the potencies of these cancers metastasis. And then we can go for the genotyping by doing the mRNA expression of these CTCs. We can do the DNA sequencing for confirmation, or NGS. If we are not able to identify the specific mutation, we can go for next generation sequencing. And then according to the genotype of that particular cancer cells, the clinician can suggest targeted therapies, and that can be given to the cancer patient. And definitely it works better because only the cells, targeted therapy kills only the cancer cells. So there is no much side effects in these therapies. So what is liquid? What are liquid biopsies are there? Liquid biopsies are non-invasive testing in which circulating tumor cells or circulating free DNA comes into circulation of blood even at early stage of cancer. Liquid biopsies can be whole blood plasma, body fluids, aspirate, or even urine samples of metastatic patient. Molecular profiling and enumeration of CTC or circulating tumor DNA concentrations can be very well predict metastatic potential of tumor cells in finding out proper prognosis and better cure for the patient. What are the advantages of liquid biopsy or the tissue biopsy, which are very important? It avoids previous methods of surgical or needle biopsies, which is very much painful and invasive. These tests capture cancer cell or DNA that tumor sheds into the blood instead of taking tissue from the tumor itself. So it is just a in vivo counterpart of a tumor inside the body. These tests permit you to take non-invasively repeated samples of cancer patient, which can be profiled for cancer gene. This is one of the very much important advantage of this liquid biopsy or circulating tumor isolation, because we can monitor the treatment of the patient at various stages of metastasis, and we can, if the drug is not responding, we can change the drug at that stage. Thus, clinician can decide specific targeted therapy as per the specific mutation to cure cancer. These tests are mostly useful when a tissue biopsy cannot easily be done, especially in the lung cancer patient. It has been very difficult. When the cancer origin site is not known, or when the drugs have stopped working and doctors are unsure what to try next to the cure this patient. Now, this situation comes in the conventional chemotherapy, because here we are really doctors. Many times I have also experienced when I was in Tata Memorial Centre Cancer Center. There are many times doctor used to say that there is no drug for this cancer patient. But this is not a situation. You can go ahead and give the therapies to this type of patient. Now, which cancer mainly require liquid biopsies? Liquid biopsies can be useful for any solid tumors like breast cancer, ovarian cancer, lung cancer, colon cancer, prostate cancer, pancreatic cancer, esophageal cancer, and now it is for the hematological malignancy also. From 2021, I think some of the references they are using liquid biopsies or circulating tumor cells, or circulating tumor DNA for evaluating hematological malignancies, because even the bone marrow aspiration is also a highly painful and invasive technology. Now, what can we do with the liquid biopsy? Liquid biopsy, various parameters we can do. We can isolate the circulating tumor cells and enumerate circulating tumor cells to know the potency of this thing. We can profiling of circulating tumor cell for various genes, which will give the targeted therapy for the patient's treatment. Then isolating circulating tumor cell free DNA. This we are not catching up. Only I will read out profiling of circulating tumor cell free DNA for various genes, exosome analysis, and mRNA analysis, which is my later part of my work, present work. Now, circulating tumor cells, therefore, circulating tumor cells enumeration and molecular profiling is a new avenue for diagnosis and treatment of metastatic cancer. Coming to the circulating tumor cells, what is circulating tumor cells? Circulating tumor cells are nothing but the tumor cells which are shaded from the primary tumor, and comes to the blood circulation. They are very large cell size, having spikes on this large nucleus and clear cytoplasm. So, and they anchored with the other side, give the secondary tumors. So, targeting these cells are very, very important if you want to cure the cancer in more easily way. So, this is a very important aspect. The circulating tumor cells were observed for the first time in 1869 in the blood of man with a metastatic cancer by Professor Thomas Ashworth. After many, many years, now the circulating tumor cells are coming to the picture in the cancer medicine. CTC appears at early stage of cancer in bloodstream before they metastasize in various part of the. This is one of the very important findings, that because as you say that the cancer is a multi-stage process. So if we get the CTC at early stage, that means the invagination may start soon, so we can take a proper care of this patient. Now, what are the significance of circulating tumor cells in cancer therapeutic? Detection of circulating cells, tumor cell is non-invasive process, and thus is it an alternative for invasive biopsy. Presence of CTC is a suggestive of minimum invasive cancer. Understanding profiling and mutational gene changes in CTC can provide suitable targeted therapy. Fewer number of CTC is indicative of longer survival time, whereas more number of CTCs is indicative of less survival time. So analysis of CTC can save a patient from worsening the condition with unsuitable medication. Properties of CTC. CTCs are present in very small number. It is between 5 to 50 per 10 ml of blood, which is our finding also. And because of the small number, you need to have a very precise method of isolating these cells. CTCs are big in size. You see on the right hand side the picture, which actually I have taken in the phase contrast microscope during my work. And you can see it is a large cells having a large nucleus spike-like surface, which are used for adhering to the blood capillary wall. CTC clog the capillary due to their large size. Sometimes they clog the blood capillary. CTC undergo complete epithelial to mesenchymal transition. This is one of the important properties of the circulating tumor cells. Once they go to the epithelial mesenchymal channel, they are more aggressive and they are more resistant to the drug. And that the clinician has to take into the consideration while giving the treatment to the patient. CTC EMT phenotype can be reverted back to the mesenchymal to epithelial transition. These are the reversible changes. CTC gives biological insight of cancer conditions, progression, and treatment prediction. Importance of circulating tumor cell in clinical medicine. Now, if you see that early detection of CTC in blood sample can help to generate faster and better remission. CTC can form the constitutional basis of tumor staging. CTC can help in identifying drug targets and save patient by predicting use of unsuitable medication. The toxicity has been completely reduced because of the CTC identification and genotype of this CTC. CTC can help in selection of secondary treatment option where the patient has failed to respond to the first line treatment. It has been several experiments shown that after chemotherapy, CTC counts still remain the high. So you have to really give little bit more vigorous dose of chemotherapy to kill those CTC cells. So treatment protocol can be monitored by using the CTC enumeration as well as the molecular profiling. CTC gives us hints and speak about the patient population that may be present in the tumor. CTC holds critical information about the tumor and its characteristic. Isolation and enumeration methods of circulating tumor cells. How we can isolate? There are various methods. Today we are discussing. Before that, we'll discuss what are the difficulties in isolating of circulating tumor cells from liquid biopsy. Very low number in blood circulation. We already discussed that. And because of the low number, you need to have the very specific method for isolating this CTC. Clogging of blood capillary due to the large size of CTC. Thus, the count obtained is not correct because there is not a regular flow of CTC sometimes. So you have to do all this work is mainly the qualitative work and not the quantitative. CTC are not present homogeneously in the circulation. Large size, CTC shed discontinuously. It is not a continuous process. CTC appearance in the bloodstream is not a continuous process. Many hurdles in CTC isolation process, and therefore people have still trying to establish the method for CTC isolation by various methods. Now we'll discuss some of the methods which are available. Isolating of circulating tumor cell by Ficol gradient method. This is the method I use along with my culture method. Now here, if you see that, take the blood, metastatic blood cancer patient, take a whole plasma collection, isolate CTC by selection of any method. I have used the Fifer gradient and use those cells in culture along with the growth medium, allow them to grow for about 15 to 20 days. And then we got isolated CTC. We can see them as according to their shapes, and they can be enumerated individually in the 10 ml of blood. We can do the phase contrast pictures. We can do the immunofluorescence microscopy. I have also done the gene expression RT-PCR genes for some of the things, which I am coming for the next of my presentation. I did the DNA sequencing for confirmation of their gene and NDS for unknown mutation, which I could not do because that time NGS was not much in population, and selection of the targeted therapy or immunotherapy. So you can have all these things done by Fifer gradient method, which is a very simple method and which is less costly, doesn't require much instrumentation. Now, second method, if you see, is the immunomagnetic antibody-based method. This is a FDA approval cell search kit method, which is already patented, and it has been introduced in 2004. This cell search kit has been extensively used by several investigators all over the world. However, this generally cell kit contains use of IPCAM antibodies to isolate CTCs. They are using IPCAM antibodies which are present in the CTCs. Then CD44 eliminate lymphocytes. Now they are eliminating the lymphocyte at the initial stage by using the CD44, which is expressed by the lymphocytes. Glycophosphorin to eliminate the erythrocytes. We can also eliminate erythrocytes by just sedimentation. Most of the erythrocytes can be sedimented fast, and you can take upper whole plasma. This way, I have done in my work. I have just done the sedimentation, general sedimentation for half an hour. We can get a good amount of CTC without RBCs. CTC undergone EMT transition cannot be detected, and a small number of such population may require. Now, using this FDA cell search kit, this is one of the drawbacks, that when the cells go at epithelial mesenchymal transition, they generally lose the IPCAM protein, surface protein, and therefore this particular kit cannot catch up these cells which are under EMT transition. So need for further improvement. This method is presently used for isolation and enumeration of CTC. Now, another method, the gaps in the cell search technology to isolate CTC from metastatic cancer. Now, some of the drawbacks are there, but they must have looked into this so far. The cell search kit is mainly based on selection of CTC on the basis of their two surface marker protein. One is IPCAM and another one is cytokeratin. Although it has been proved greatly useful and reproducible, it may, however, limit the selection of CTC from highly metastatic patient due to the down-regulation of IPCAM in this type of patient. Because when there is a highly metastatic patient, most of the CTCs undergo epithelial mesenchymal transition, and there is a problem in enumeration of these CTCs by this method. Lenidio et al. 2011 has shown that CTC which have undergone EMT will show downregulation of epithelial markers including IPCAM, and therefore this technology will miss this CTC. Similarly, in some cases, it has been shown that HER2 positive metastatic breast cancer cells show IPCAM negative CTCs. So when there is even they have shown the correlation between the HER2 expression and IPCAM negative CTC. So hence, IPCAM independent method could fetch an increase in number of CTC in those cancer patients and need further improvement in this direction. Now, next method is cell size bead separation, isolation by size of epithelial tumor tissues called INSET. This is done on Metascale device is used for this study. Cell size is used for sorting. Specialized filters are used for filtration. Collection is observed over the filter membrane. Metacell device has been used for size-based separation CTC compared to the other blood cells for CTC enrichment from whole blood. Here is the protocol. You can see the 10 ml blood mixed with acid buffer. Metacell device give the filtration, and that filters we can isolate the CTCs for various membrane. Along with these CTCs can be detached and transferred to the 6-well plate for circulating or directly used for the characters of CTC by immunofluorescence or histopathological staining. So this method is very much useful because this gives the cells, live cells, without tagging with any antibodies, so we can culture these cells. Microfluidic method for circulation of tumor cells proposed. Actually, I have proposed this particular prototype. We have done it, but because of the COVID pandemic, we could not go further in this experimentation. But this is a very simple device which I have planned is that there is an inset for whole blood plasma, inlet for the whole blood plasma, inlet for the buffer which goes, and outlet for the RBCs according to the size. Small size RBCs are extracted, then little larger size the WBCs are comes, and CTCs are almost by the 50 micron, which size they fractionated at the last moment. So this is an antibody-free system. It is a cell size based sorting accomplished by using microfluidic technology. So this is also one of the very important technology which give the cell type live cells without any tagging with any antibody. Enumeration of circulating tumor cell in cancer therapeutics. Some of the use of circulating tumor cell in cancer therapies has been given in the next slide. Last several years, Vidart et al. 2013 have been studying circulating tumor cells for evaluation of diagnosis and therapy in metastatic cancer. Now, Vidart, I have taken the reference of Vidart because so far, the Vidart from Paris, he has almost published 42 papers in this area, and he has clearly shown that circulating tumor cells is one of the very important parameters for diagnosis and therapy of metastatic cancer. There is a direct correlation of number of CTC with disease progression and survival has been reported in many cases. It has been so far established that for patients having more CTCs, there are less chances of survival. When there are more than 80 here, it has been shown that patients having 80 or more CTCs in 7.5 ml blood, because most of them use the cell search kit, which requires 7.5 ml blood, have a maximum 1 year survival. Hence, it has been shown that CTC enumeration is very important early and metastatic cancer patient. Analysis of CTC enumeration in cancer therapeutic. Now, chemotherapy is one of the standard mode of treatment for metastatic cancer. During the course of this therapy, CTC values are determined before and after round of therapy. In most of the cases with non-metastatic state breast cancer, reduction in number of CTCs is observed after the first round of chemotherapy. But it is also noted that CTC had the tendency to realize resistance to the therapy. Hence, it is suggested that the procedure of increasing dose should be arranged in progressing around of the chemotherapy. Here we can clinician get a clue if there are still after therapy also, if there are still the circulating tumor cells found, they can certainly go for the higher therapy or they can change the therapy. Peter et al. 2000 has revealed that a patient who had more than 80 CTC in 7.4 died within 1 year from diagnosis of metastatic of the disease. So he is also a metastatic breast cancer patient, and so if once the clinician know that the patient has high CTC, they can take more precaution in managing these diseases more preferably by using CTC enumeration. Smirch et al. 2014 has observed in metastatic breast cancer multicentric project that CTC continue to remain detected after the first round of therapy in some patients of breast cancer with rapid progression of the disease to metastasis. Therefore, it has been suggested that in such cases, it can be ideal to offer some alternative treatment with some novel therapeutic agent rather than continue with the same therapy. This is the same what we have just now talked, that CTC really give us an understanding to give the better treatment for the patient. Molecular profiling of circulating tumor cell. This is also one of the very important part. Now, since Human Genome Project, the molecular diagnostic has been initiated in cancer diagnostic. Great advantage of Human Genome Project. This is done by Francis Coding from NIH director, who is really proud to say about this thing. The completion of Human Genome Projects revolutionized cancer diagnostic and treatment by development of newer, cheaper, and more reliable methods of gene expression analysis. By understanding molecular profile of individual cancer patient, allow clinicians to determine origin of cancer, its potential for metastasis, its specific drug responsibility, and probability of its recurrence. So this whole thing we can do by using the molecular diagnostic. Various technology has been introduced. This technology now, I think everyone knows, since COVID pandemic, these technologies are now known to everyone. These are PCR, real-time PCR, microarray analysis, DNA sequencing, next generation, and fluorescence in situ evidence. These are the technology. We are not going in detail about this. This technology are very much useful in understanding the genotype of this cancer cells or circulating tumor cells. Enumeration on molecular profiling of circulating in cancer therapeutic. Vidal et al. 2010 reviewed clinical application of CTC in metastatic breast cancer therapy. They have assessed the use of HER2 treatment in HER2 negative breast cancer patients, which are selected on the basis of CTC dissection. They have used the cell search kits for this study. Now here the case was that they found that the patient is HER2 negative when they have done this tumor biopsies. But when they have extracted the CTC, they found that CTCs are HER2 positive, and then they have used HER2 positive treatment to treat this patient, and there was a response to the treatment. So this is one of the advantage of CTC genotyping. Vidal et al. 2016 have also studied clinical utility of CTC in metastatic breast cancer. They have mainly focused on first and second line treatment for this breast cancer patient. Rack et al. 2004 concluded their study that there is independent prognostic relevance of CTC determination both before and after adjuvant therapy in patients with primary breast cancer. They have studied more than 2,000 breast cancer patients before and after therapy. They also have used cell search kit for the CTC isolation and enumeration. Smirch et al. 2014 have confirmed the prognostic significance of CTC in patient with metastatic breast cancer received for this therapy rather than continuing with the same therapy. In case of molecular profiling, there are various molecular profiling gives an understanding of genotype of CTC. There are various genes involved: MMP1, MMP2, MMP9, VEGF, COX2, ICAM1, adhesion molecules, E-cadherin. These all genes I have been studied in my CTC. That I'm coming there. Role of cytokines and chemokines such as IL-6, TGF-alpha, CXCR4, and epithelial markers such as keratin 18, 19, 20, 21 are the major markers. Other genes involved in the fibronectin extracellular protein like CD44 and CD33 genes, which are mainly the markers for the cancer stem cells. And CTC is nothing but the cancer stem cells, which really gives a resistance to the therapy. Other markers like researchers have shown that keratin 19 and p53 mutations are mostly found in CTC derived from triple negative breast cancer patient, and it is one of the factors for progression of this disease to the triple negative. It has also been shown that breast cancer patients who are express keratin 19 and RB2 genes showed poor survival. EMT changes have been one of the critical properties, and these are the markers like ET5, Notch1, SNEL, TJ-beta, JEB1, JEB2. So if we get this type of genes into the circulating tumor cells, we can say that there is a EMT transition. The mutational and transitional changes which take place in the CTC make them gain an aggressive behaviors which in turn help them to break apart the basement membrane and disseminate it from the tumor, because this gives the proteolytic activity and broken the basement membrane and invagination process start. Importance of CTC profiling diagnosis and therapy breast cancer. For 2002, I show that HER2 negative breast cancer patient, HER2 positive CTCs are identified. Same thing I already explained to you. This led to the consideration of revision in the ongoing treatment of the disease. Trastuzumab-based therapy is applied to this patient with HER2 positive CTC and HER2 primary tumors. This study has shown that CTC can help in identifying the changing course of disease well before time. Thus, CTC do hold the potential to represent a metastatic state of breast cancer. Now, coming to the enumeration and molecular profiling of circulating tumor cell in diagnosis therapy, this is my work in my laboratory. I have been working in the circulating tumor cell recovery for last 7 years, and I have published 3 papers. I have also made the guest. I was a guest editor for just Journal of Cancer Metastatic and Treatment Journal, and we have published 9 papers in this. So these are the major publications in circulating tumor cell from my laboratory. If you see my protocol for isolation, enumeration, and molecular profiling of circulating tumor cells, this is my actually protocol. I have taken the metastatic cancer patient, collection blood and separation of whole plasma by Fecal gradient, and then this Fecal gradient I have used the tissue culture method to separate CTCs. I have manually counted CTCs, enumerate CTCs, and this count was very well correlated with the potency of the metastatic patient history. And then we did the molecular profiling of CTC, and this much portion was actually approved for this, because going for the targeted therapy and monitoring response to the therapy was the further clinical studies which needs a special permission from the government of India, and that's I was waiting for the next period. But due to the COVID pandemic, this project has not been gone further so far. Now, if you see my results, this is the tissue cultures cells in the the CTC separated for the cultures. You can see this very well. CTCs have a different structure. They are large in size. They have spike. This is a higher magnification. You see the clearly spike, which I have already shown you the picture, and this is a Chinese staining. So this we can count and enumerate. Profiling also by this is I have done encourage independent growth so that they have the metastatic potentiality, and you can see they are growing on the soft agar and showing the proteolytic enzyme ring. These are highly proteolytic enzymes which break the basement membrane, and more metastasis has been occurs. This shows the highly metastatic potentiality of these cells. Coming to the molecular profiling, I have done various genes, almost K18, CD44, CD133, CXCR4, NOSH2. These are mainly the cancer stem cell genes, and they are all expressed by the circulating tumor cells, whereas this is the genes which are also expressed by the CTCs from. There are 3 breast cancer patient, nasopharyngeal, 1 ovarian cancer patient, and 1 prostate cancer patient. They are highly metastatic highly metastatic cancer patients, and their expression, you can see there are breast cancer is down-regulating K19, indicating it is a very aggressive tumor. These 2 are aggressive, whereas this breast cancer has some possibility of better prognosis. So this way, if you see the MMP1, MMP2 is also expressed, MMP9 is also expressed, so metastatic gene is there. ERG1 is totally absent in this showing. Then ICAM1 is also present in some of the showing that they may be having the cluster CTC, which are the adhesion molecules attachment, individually attachment. So, and whereas some no expression showing that they are single CTCs. Then each is also most of the cells are in EMT phase. Next slide is molecular profiling circulating tumor cells. This is actually the circulating tumor cells which I have grown in the culture. They are taken for this. I have extracted RNA. I am expert in extracting RNA from small amount of cells, and we have done this. Here you can see the most of the genes which are expressed are cancer stem cell genes, and it's indicated that circulating tumor cell is one of the cancer cancer stem cell like cell which is resistant to therapy, and therefore the targeting of these cells will give better results in cure of the cancer patient in future. So in conclusion, I want to say blood testing for circulating tumor cells has emerged as one of the hottest field in cancer research. CTC detection and enumeration can serve as an early marker of response to systemic therapy, whereas their molecular characterization has strong potential to be translated to individualized targeted therapies and spare cancer patients from unnecessary and ineffective therapies. Different analytical systems for CTC detection isolation has been developed, and new area of research was the developing novel assays for CTC molecular characterization. Molecular characterization of single CTC holds considerable promise for predicting biomarker assessment to explore CTC heterogeneity. The application of extremely powerful next-generation sequencing technology in era of CTC's molecular characterization in combination with reliable single CTC isolation open new frontier for the management of cancer patient in near future. And this is my group actually. Navjit Kaur, who has written the review, which review has been so far seen by 68,000 people. It is in JCMT, Journal of Cancer Metastatic. And Kirti is the one another paper which I have published on CTC. Other staff, they are all done their PhDs. She is my PA and some staff members. And this is one of the group working on the circulating tumor cells. Thank you very much. Thank you. Any question?
Thank you, Dr. Potdar, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. We'll answer as many of your questions as we have time for. Let's get started. So our first question, Dr. Potdar, is actually a bigger question followed by a follow-up question. So the first one is, how sensitive are the CTCs in the patient collected blood sample? And the next part of that was, do you recommend any sample collection and or storage practices to ensure the circulating tumor cells are viable at the time of enumeration?
Yes. Yeah, CTCs are very sensitive actually cells, but they are stable when they are collected from the blood. CTCs are the in vivo counterpart of the growing tumors and are live cells when isolated from the blood samples. So they are stable. So there is, at least you can process within 24 hours, because most of the time my experiment was immediately I used to carry out. Regarding the second question, do you recommend any sample collections or storage practice to ensure the circulating tumor cells are viable at a time of enumeration?
Okay.
If sample collection, we generally do it by the EDTA tube containing EDTA, not heparin, because if you want to do the molecular analysis, possibly the heparin may interfere into your reaction. So mostly the EDTA is a prefer tubes for collection of the blood. And we can isolate live CTC by Ficol gradient, which I have already said, Ficol gradient method or microfluidic separation or isolate filtration method. If we want to keep CTC alive for longer time, we can culture them for longer time. They will live alive, but keep live for long time. But otherwise, these cells can be frozen in minus freezing media and kept at minus at liquid nitrogen. They may give they are live, but I have not used any of this method because I have mainly carried out the work immediately after I get the blood samples.
Okay, thank you so
Next
much.
question.
That's very helpful. Yeah. So the next question is, how does purity, recovery rate, and or viability of cells impact molecular characterization of CTCs?
It is very much, because when you are doing molecular analysis, you must have the fresh sample for analysis, So most of the when you are working on RNAs or mRNA expressions, RNA has to be extracted as early as possible. It is always advisable process CTC for molecular analysis. Else you can use the RNA letter solution, which I have not used it, but you can use the RNA letter solution in which you can keep the cells, but they are not live. If you want to keep live cells, you can freeze them under liquid nitrogen,
Okay,
and
that
extract.
makes sense.
And my study we have processed the cells immediately. That's okay.
Sure.
Did you get all of it?
Yeah, absolutely. That was great. Thank you so much.
Okay.
Good. So the next question is, what is the most common application to use the isolated CTCs?
CTCs have two major part. One is enumeration, which give the potency of the cells to metastasize, and second one is the molecular profiling, which you can use for giving the targeted therapies. So these are the two major functions of CTC in clinical medicine, cancer medicine.
Okay, excellent. The next question is, would there be any clinical value in isolation and characterization of CTCs in addition to ctDNA from liquid biopsies and solid tumors from the patient?
Yes, definitely. CTC and ctDNA are both used for clinical evaluation of metastatic cancer. CTC reflect more of the metastasis initiating cells, while the ctDNA represent more the tumor burden. Presently, clinicians are using both the methods for evaluating the metastasis and metastatic tumor burden. CTC can be used for in vivo studies also. That is one of the advantage of CTC. They are live cells, whereas tumor DNA is the DNA which is extracted from the apoptotic cells, any apoptotic cells, which is present in the blood. CTC also can evaluate the chemotherapy, whereas ctDNA only will give the genotype of the cells. So these are the major differences using the CTC and ctDNA. But both tests are very much essential and can be applied if there is any difficulties in giving the treatment to the patient.
Okay, wonderful. Thank you, Doctor. It looks like we have time for one more question. So the very last question is, what is the importance of CTC clusters?
Now, when I have done the work on CTC, I have seen almost single cell CTC. I could not get a cluster, even the cells where tumor were aggressive. But when you get the clusters of the CTC, it indicates that tumor is very much aggressive. And therefore, you have to really careful about giving the cancer therapy to the patient. Now, CTC clusters have two types. One is homotypic, and second is heterotypic. Homotypic have the CTC cells more than 2 cells or maximum is 20 cells cluster. They are similar to the single cell CTCs. Maybe their genotype also is similar. Whereas heterotype clusters, they have a combination of some of the cells, immune cells, as well as cancer-associated fibroblasts. So and if such type of clusters you get it clearly shows that the tumor is very, very aggressive and the prognosis is poor. CTC clusters are 100 times more severe, more metastasis than the single CTC in the breast cancer or any other cancer or prostate cancer. For using the technology, we have to use cell search kit also has used for the isolating CTC clusters, but there will be a 10 to 17% of the CTCs clusters can be isolated by this SIM. But there are some methods which I have explained you now. The Ficol gradient method also we can use the CTC. The best method is isolate, which is a filtration method where you can get a clusters and you can extracted these clusters in the culture dish, and you can see the cluster cells, live cells in this, and then you can do the genotyping. So microfluidic assay also can use for isolating the clusters. So clusters has specific method for isolation. But if you clinically, if you see when you get a cluster CTCs, that means you are getting a very unfavorable clinical outcome, and therefore in that patients, definitely for studying those patients are very important if our views are concerned to cure the patient. There is one method which is reported by Rolet et al. 2020, latest one, that biosurface CTC technology. This isolate the CTC clusters from the whole blood of the cells. So this is about the CTC clusters, I can say.
Wow, that's very interesting. Thank you so much. And thank you, Dr. Potdar. Do you have any final comments for our audience?
I just want to say that I have just my experience is concerned in the cancer research and therapeutic. Still, this particular technology has not been used by many clinicians, and they only some certain hospitals. So I personally feel that this technology should be used in each and every cancer set of hospitals, because this is a very important technology which reduces the doses of the chemotherapy, which reduces the toxicity treatment, and patient has better survival than doing the conventional therapy. You can monitor the therapy. Doctor will get insight of this full idea about what is the tumor, inside tumor is doing. Only looking at the certain specific area of the cancer. Suppose I have seen that many times we see that there is a regression of the tumor after giving the targeted therapies, but it appears after 1 year relapse, because there are cells inside. There are some silent cells which are present in this tumor. Even you see the CT scan that you see that there is a regression of tumor, but a small part of the tumor remains. They are the cancer stem cells, and these cancer stem cells again regrow, and these circulating tumor cells are nothing but the cancer stem cells which we have shown with our experiment. So I personally feel that this particular technology should be introduced in almost all hospitals for the cancer therapy, and the cost wise it should be kept for the patient affordable cost, so that every patient can afford this. Thank you very much.
Absolutely. Thank you again, Dr. Potdar, for your time today and your important research. We would like to also thank Labroots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speaker via the contact information that you provided at the time of registration. This webcast can be viewed on demand. Labroots will alert you by email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.
Goodbye. Thank you very much.